One of the largest mass poisonings in human history has been happening since the 70s in Bangladesh. And it all started because, to combat waterborne diseases, Bangladesh promoted the use of deep tube wells for drinking water. These were considered to be safe. And then in the 90s, scientists and health workers began noticing symptoms of arsenic poisoning, such as skin lesions, cancers, and organ damage. Tests confirmed high arsenic levels in the groundwater across the country, a poison that had already begun its slow, insidious attack on their skin, their nerves, their DNA. But what happened? And what is arsenic exactly? Initially, these deep tube wells were promoted by international aid organizations to reduce deaths from contaminated surface water. It was a well-intentioned solution, until it was discovered that these wells were contaminated with naturally occurring arsenic. And today, millions of people in Bangladesh, India, China, and even parts of the United States are exposed to dangerously high levels of arsenic in their drinking water. This crisis has been called the largest mass poisoning in human history. But what is it exactly, and how does it contaminate our waters? Arsenic is a naturally occurring metalloid found in the Earth's crust. It exists in two main forms: organic arsenic, which is relatively harmless, and inorganic arsenic, which is highly toxic and contaminates millions of wells worldwide. It enters drinking water through two main routes: natural erosion of arsenic-rich rocks and human activities, for example, pesticides, mining, and industrial waste. And this leaches arsenic into our water supplies. The most dangerous forms are arsenite and arsenate, and are often found in these contaminated water sources. But the real danger is that you won't even know that you're being poisoned. Unlike lead or mercury, arsenic has no taste, no smell, and no immediate symptoms. People can consume contaminated water for years before realizing the damage: cancer, organ failure, and neurological disorders. This is why arsenic is often called a silent killer. Once arsenic enters the body, it begins to disrupt essential biological processes, leading to widespread toxicity. Arsenate, a major form of arsenic in groundwater, structurally resembles phosphate, allowing it to enter cells through phosphate transporters. But unlike phosphate, arsenate disrupts energy production. Normally, glyceraldehyde 3-phosphate dehydrogenase helps generate ATP, but arsenate replaces phosphate, forming an unstable intermediate, 1-arseno-3-phosphoglycerate. This breaks down prematurely, preventing ATP formation and starving the cell of energy. Simultaneously, arsenate reforms during this reaction, allowing continuous ATP depletion. Arsenite, an even more toxic form, disrupts cells in multiple ways. It inhibits the citric acid cycle by binding pyruvate dehydrogenase, blocking acetyl-CoA production and reducing cellular energy. It also triggers oxidative stress by generating reactive oxygen species through the Fenton reaction. This causes DNA damage and depletes protective antioxidants like glutathione. Beyond energy disruption and oxidative damage, arsenite affects cell signaling. It overstimulates the sphingosine 1-phosphate receptor, activating the RAS-RAF pathway. This uncontrolled signaling drives excessive cell proliferation, increasing cancer risks. To understand how arsenic affects cells at a molecular level, toxicogenomics is used, analyzing how gene expression changes in response to toxicants. In a study using iPSC-derived kidney cells, the mRNA expression after arsenic exposure was examined. Within just 5 hours of exposure to 10 micromolar sodium arsenite, the metal stress pathway was activated, depicted here in red. This pathway plays a crucial role in working to detoxify metals and maintain balance. Key players are metallothioneins, tiny but powerful proteins that normally bind to metals, such as zinc and cadmium, to neutralize their toxicity. However, arsenic presents a unique problem. While it induces the metal stress response, metallothioneins cannot actually bind arsenic. This means that despite the strong induction of MT genes, which maintain high levels of expression throughout the 24-hour period, the damage caused by arsenic continues unabated because it remains unneutralized. But arsenic's activity doesn't stop there. It also cranks up oxidative stress. That's where the NRF2 pathway comes in, setting off alarms in the cell, activating the HMOX1 gene. This gene produces heme oxygenase 1, which breaks down into biliverdin and then into bilirubin, a natural antioxidant. Basically, your cells scramble to protect themselves before arsenic-induced chaos takes over. Now, what happens if you're exposed to arsenic for days, weeks, or longer? Researchers checked Hep G2 liver cells after 10 and 20 days of exposure, and things only got worse. Gene expression went haywire. The metal stress pathway gene expression ramped up to 4 times their normal levels for higher arsenic concentrations and doubled for lower concentrations. While the HMOX1 gene skyrocketed by 5-fold at higher arsenic concentrations. So what does this all mean? Well, at first, your cells try to fight back with these protective mechanisms, but over time, arsenic overwhelms their defenses, leading to oxidative damage, cellular dysfunction, and eventually disease. Chronic arsenic exposure is a slow and silent process. By the time symptoms appear, the damage is well underway. At first, it's easy to miss. A few dark spots on the skin, maybe some rough patches on your arms and feet, but nothing too alarming. However, these early signs of hyperpigmentation and keratosis are actually warning signals that arsenic is accumulating. Some people start dropping objects or struggling with fine motor tasks, but they often don't connect it to arsenic exposure until it's too late. Then, arsenic slowly infiltrates the nervous system, leading to peripheral neuropathy, a numbness, tingling, and weakness that makes even simple movements feel off. Then comes the real danger: cancer. Arsenic is a group 1 carcinogen, meaning we know for certain it causes cancer in humans, and it doesn't just target one organ. It goes after the skin, the lungs, the bladder, and the liver. This mechanism behind these symptoms, like mentioned before, is a brutal mix of oxidative stress caused by the Fenton reaction, which causes DNA mutations and a promotion of key tumor growth pathways, like the RAS-RAF pathway. Long-term exposure doesn't just increase the risk, it almost guarantees a risk of higher chance of malignancy. Because arsenic concentration is so widespread, regulatory agencies have implemented strict measures to control exposure and protect public health. First, the European Chemicals Agencies, or ECHA. Their job is to classify arsenic as a carcinogen and reproductive toxin under REACH regulations. This means that its use is restricted in industries and workplaces, ensuring that exposure is minimized wherever possible. But ECHA does more than just label arsenic as dangerous. They also set exposure limits, influencing environmental and food safety laws. Next, we have the European Food Safety Authority, EFSA. This agency focuses specifically on arsenic in food and drinking water. They conduct toxicological studies and track dietary exposure across Europe. And surprisingly, one of the biggest culprits is rice, because rice absorbs arsenic more readily than other crops. EFSA keeps a close eye on contamination levels. Their most recent 2024 report reinforced what scientists have warned for years: chronic arsenic exposure increases cancer risk, which has pushed for stricter food safety regulation. Then there's the European Commission. This is where research turns into action. Based on ECHA's chemical safety data and EFSA's food risk assessments, the Commission creates legally binding EU regulations. A great example? The 2023/465 regulation, which lowered arsenic limits in rice, infant food, and fruit juices. These aren't just guidelines, they're laws, and the Commission ensures that every EU member state enforces them. And finally, on a global scale, we have the World Health Organization. and until recently, almost every country was a part of this organization. Their guideline influenced regulations worldwide, including the 10 microgram per liter safety standard for arsenic in drinking water. WHO also leads research on arsenic-related diseases, like cancer, cardiovascular disease, and developmental disorders, shaping international policies that protect communities everywhere. Computational models play a crucial role in assessing the toxicity of arsenic and its compounds, helping regulatory agencies make informed decisions about public safety. By analyzing chemical structures and predicting biological effects, these tools provide a faster, more cost-effective alternative to traditional laboratory testing. One of the most widely used methods is quantitative structure-activity relationship, QSAR. QSAR models use mathematical algorithms to find patterns between a chemical's molecular structure and its toxicological effects. For arsenic, QSAR can predict carcinogenicity, cytotoxicity, and environmental persistence based on molecular descriptors such as atomic charges, bond types, and electronic properties. This allows regulators to assess risks before new arsenic-containing compounds are widely used. Another important computational method is read-across, which estimates the toxicity of an untested chemical by comparing it to similar substances with known toxicological profiles. This approach is especially useful for arsenic compounds that lack extensive experimental data, such as certain organic arsenicals and industrial byproducts. Read-across is widely used in regulatory frameworks like the EU's REACH regulation, which encourages non-animal testing methods for chemical safety assessment. But what about the future? Can we fight arsenic? Are we doomed? Well, regulatory agencies are reassessing their arsenic regulations. Stricter maximum allowable levels in drinking water and food products are being considered, with a particular focus on vulnerable populations, including children and pregnant women. As mentioned earlier, the European Commission already recently reduced the permissible arsenic levels in food, especially in rice and baby food, and similar adjustments are being considered in regions all around the world. Scientists are increasingly turning to computational models, AI-driven predictive tools, and machine learning algorithms to assess arsenic toxicity more efficiently. These models help predict potential risks, allowing for proactive policy adjustments before health crises emerge. Monitoring programs are being expanded to track arsenic levels. Countries with high exposure risks, such as Bangladesh, India, and parts of the US, are investing in more frequent testing in drinking water and soil contamination levels. Chronic arsenic exposure likely remain a public health concern for a long time, particularly in countries where natural arsenic contamination in groundwater is just prevalent. However, ongoing scientific advancements, stricter regulations, and global efforts, the long-term health impact of arsenic poisoning can be mitigated. The key will be sustained monitoring, innovation in detection and filtration technologies, and a stronger enforcement of regulatory standards to protect future generations and fight arsenic contamination.